Hello, everyone. This is the first topic of cytogenetics, and I'm here to introduce to you the study of cytogenetics. The learning objective of this lecture is to explain the general concepts and principles related to cytogenetics, to differentiate terms commonly employed in the study of genes, and identify the pioneers in cytogenetics and their contribution, enumerate the three main areas of genetics and their areas or field. These are the references for this topic. And first is let's define cytogenetics. Cytogenetics is a branch of genetics that deals with the study of inheritance in relation to the structure and function of chromosomes. Now, majority of the topics that we'll be covering in cytogenetics, and specifically the clinical conditions that are mentioned in cytogenetics, are chromosomal aberrations or chromosomal abnormality. But before understanding about the chromosomes, we need to understand first what is the composition of that chromosome and how that chromosome came to be. So this shows you an illustration of the cell. Now, we know for a fact that a cell contains a nucleus, and in human cells, we have 23 pairs of chromosomes with a total of 46 chromosomes. And inside the nucleus, chromosomes are there because it serves as basically the chromosomes are just a wrap around of DNA. It's just a condensation. It's a condensed structure of the DNA, and the purpose why we need to condense the DNA because if you would stretch the entire length of DNA, it would be around 2 meters long. Now, in order for that 2-meter long structure to be inserted into that very microscopic structure of the nucleus, it has to be arranged in a way that it is very compact and condensed. That's why we have the chromosomes. Now, cytogenetics would involve learning about nucleic acids. That's why we have topics in nucleic acid as part of cytogenetics. So we study the biochemistry of nucleic acid as well as the structure of deoxyribonucleic acids. We also be studying on the structure and architecture of chromosomes and the genes, how genes are placed in chromosomes. We also need to understand about the cell cycle, the Mendelian patterns of inheritance, chromosomal abnormalities and diseases, as well as cytogenetic techniques. So these are the things that we'll be covering for cytogenetics. Now let us define some important terminologies. Now, I want you to remember these terms because these terms are frequently used in cytogenetics. When we say character, this is the heritable feature that varies among individuals. So example of a character is, for in the illustration shown on the right, is flower color. Or if it was applied in humans, eye color. Or hair color, skin color. That is character. When we say trait, it pertains to each variant of a character. Now, for example, if we discuss about the traits of a certain character like flower, then the traits are purple or white. Now, if we apply that to human, if we say a character of eye color, then the traits can be brown, green, or blue eyes. True breeding refers to parents that would produce an offspring that would carry the same phenotype, meaning the parents and their offsprings, meaning sons or daughters, would have the same physical expression or visible expression. Hybridization refers to mating of or crossing of two varieties. P stands for parental generation, which means true breeding parents. F1 refers to the first filial generation, and these are the offsprings of the parental cross. F2 is the second filial generation. These are the offsprings that allow to pollinate or cross to one another. The term genotype is a genetic composition of an organism, while phenotype is the visible expression of information contained in a gene. So you will always encounter these terms genotype and phenotype. Locus, which is the singular form, the plural form is loci. These are specific fixed position on a chromosome where a particular gene is located. When you say allele is an alternative form of a gene. A homozygous refers to an organism having a pair of identical allele at a given locus at a given chromosome. When we say heterozygous, it means that an organism has two different alleles at a given locus. Okay? When you say genes, these are nucleotide sequence coding a polypeptide sequence which makes an enzyme or structural protein. Now again, genes, these are just series or patterns or the sequence of the DNA that will code for a specific protein. Now this illustration shows you several locus or loci. So this is the in chromosome number 2 you see here. This is chromosome number 2 because this is the numbering of the chromosome, and you can see here that at this specific portion of the chromosome is where you can find the gene that will code for producing brown eyes. Okay, and this specific locus is where you can find the genes that would code for the body. Okay, now just a brief review of the history of genetics or cytogenetics. Before 1860, the discovery of the cell and nucleus was actually originated from the beliefs of Hippocrates. So the general beliefs of Hippocratic school persisted, and in this belief that the gametes of an organism would assemble into the individual. Nicholas Hartsocker created the homunculus. Now, homunculus is the one that we use in the brain, wherein we can identify the specific portion of the brain that is responsible for motor movement. But Nicholas Hartsocker created a homunculus in which a miniature human carried in the head of a sperm. William Harvey, he discussed about the theory of epigenesis. Robert Hooke, at 1665, observed the cell structure under the light microscope. Anton van Leeuwenhoek was a master lens maker, and he made the single lens microscope. So he created the single lens microscope and therefore paved the way to allow us to study this microscopic structure called the cells. Jan Purkinje was the first scientist that described the nucleus within the cell. Robert Brown was a botanist, and he coined the term nucleus. He also discussed the Brownian motion of microscopic particles, and he differentiated or he discovered the gymnosperms and the angiosperms. Hugo von Moll, he was the one who discovered mitosis in a cell, and Rudolf Virchow summarized the concept of cell theory. And in this cell theory, all cells come from pre-existing cells. Now, during 1860 to the 1900s, we have now Gregor Mendel. Gregor Mendel was an Austrian monk. He discovered the classical hybridization experiments with pea plants. And allowing us to understand the statistical patterns of heritable phenotypes. Oscar Hertwig was an embryologist, and he discovered that a zygote is formed when there's fusion of sperm and egg. Walter Fleming was a physician and newly synthesized aniline dyes to view and describe chromosomes and the way they move during mitosis. Heinrich Waldeyer was the first scientist to use the term chromosome. Theodore Boveri, Karl Rabel, and Edward Beneden hypothesized that chromosomes are individual structures with continuity from one generation. August Weismann, he discovered that inheritance of traits is based exclusively in nucleus, and he also described about meiosis. Hertwig and Boveri described the process of meiosis in detail. During the 1900s to 1944, we now have the emergence of the chromosome theory and sex linkage. Thomas Hunt Morgan introduced Drosophila melanogaster as a model genetic system. The Drosophila melanogaster is actually a common fruit fly. Walter Sutton explained Mendel's rules of inheritance, while Nettie Maria Stevens investigated sex determination in mealworms, and she also described about X and Y chromosomes. Thomas Hunt Morgan discovered that there are certain genes that produces white eyes, yellow body, and miniature wings in Drosophila, and these genes are located in the X chromosome. And these findings would now result into the discovery of the X or the sex-linked genes. Alfred Stuttervant used Drosophila to create the first genetic map. Calvin Bridges, working with Morgan, described non-disjunction of Drosophila sex chromosome. Louis Stadler and Hermann Mueller, he discovered that genes can be mutated artificially by X-rays. William Bateson, he used the terms F1, F2, homozygote, heterozygote, and allelomorph and created the term genetics. Wilhelm Johansen described the terms phenotype, genotype, and gene, while Ronald Fisher, Sewell Wright, John B.S. Haldane, they founded the algebraic understanding of the process of evolution. Salvador Luria and Max Delbruck demonstrated that bacteria have genetic systems and phenotypes that could be studied. From 1944 to present, we have now the emergence of molecular genetics, and in this time, the nucleic acids would come into place. Oswald Avery, Alfred Hershey, and Martha Chase, they described that DNA was genetic material. James Watson and Francis Crick worked out the structure of DNA. They created the model of the DNA. Werner Arbor, Hamilton Smith, and Daniel Nathans described restriction endonuclease as the servant with the scissors, and they won a Nobel Prize in 1978. We will discuss further about restriction endonucleases when we go to molecular biology. Paul Berg was the first to construct a recombinant DNA molecule containing parts of DNA from different species. And in 1995, it was the Haemophilus influenzae was the first organism to have its complete genome sequence. In 1996, the first mammal was cloned, which is Dolly by Ian Wilmot. And in 1997, the first transgenic cow, or Rosie, can express human alpha-lactalbumin protein in her milk. In 2001, the human genome was sequenced and published. And in September 2007, there are 634 different organisms that have had their complete genome sequenced and published. Now, the general areas of cytogenetics include classical genetics, molecular genetics, and evolutionary genetics. For classical genetics, it is concerned with genes, mutations, and phenotypes. It deals with the arrangement of genes on the chromosomes, methods that generate mutations, and various patterns of inheritance. Gregor Mendel published the basic rules of inheritance. He correctly hypothesized that traits, such as pod colors, were controlled by genetic elements. He also predicted that adult organisms have two copies of each gene, which we call diploid states, and gametes receive just one of these copies, called haploid state. Gregor Mendel also formulated two general laws of inheritance: law of segregation and law of independent assortment. Classical genetics also deals with the chromosomal theory of inheritance. In this chromosomal theory of inheritance, chromosomes which contains genes are the carriers of genetic material. It is George Beadle and Edward Tatum suggested that one gene encodes one enzyme. We have what we call one gene encodes one polypeptide. The application of classical genetics include pedigree analysis and karyotyping, in which we will be covering in detail. For molecular genetics, which is actually one of the first parts of cytogenetics topic that will be discussing, is we focus on the central dogma of life. Now, the central dogma of life, or the central dogma of molecular biology, states that DNA has the capacity to replicate itself, and that DNA can be transcribed to form mRNA, and mRNA can be translated to make a protein. Therefore, in the central dogma of molecular biology, DNA makes RNA makes protein. Now, molecular genetics, it deals with the study of the structure, replication, and expression of the genetic material and of the expressed protein. The most important molecule in molecular genetics is the deoxyribonucleic acid. It is the genetic material for all cellular organisms. It is a double helical molecule and it is antiparallel. We have bases such as adenine, thymine, guanine, and cytosine. In terms of DNA replication, double helix unzips, resulting in two double helices, exactly like the first. The DNA polymerase enzyme performs DNA replication, but we'll be covering more about this in detail as we go to the central dogma of molecular biology. So this table shows you the genetic code dictionary of RNA, and it shows that the sequence of 3 codons would code for a specific amino acid. So in molecular genetics, we have the term transcription, and as what I've mentioned, transcription is the conversion of DNA to RNA. And please do take note that RNA is different from DNA in that RNA contains ribose and it has uracil rather than thymine, and it is usually single-stranded. It is transcribed by DNA by the enzyme RNA polymerase. There are types of RNA, and mRNA is the one directly involved in the transcription process. During translation, it is the conversion of mRNA to protein. We'll be discussing that in further when we go to the central dogma of molecular biology. The applications of molecular genetics include recombinant DNA technology, and it actually began with the discovery of restriction endonucleases. It utilizes plasmid, small, circular, extrachromosomal DNA units found in some bacteria. PCR, or polymerase chain reaction, amplifies a desired DNA sequence, which is an application of recombinant technology, and this is able to locate disease-causing genes as well as suggesting potential treatment. Other applications of molecular genetics include genetic engineering, genomics, and transcriptomics. For evolutionary genetics, it studies the mechanism that results in changes in allele frequencies in population over time and the consequences of those changes. So in evolutionary genetics, we deal with the larger population. Charles Darwin described the concept of evolution as a result of natural selection. Godfrey Hardy and Wilhelm Weinberg formulated the Hardy-Weinberg equilibrium. It is used as a baseline for comparing a population to see whether any evolutionary processes are occurring. The Hardy-Weinberg equation was used to relate allele frequencies and genotype frequencies. It applies to a gene in a diploid species that is found in only two alleles. An application of this equation is the field of sociobiology, in which social behavior is under genetic control and is acted on by natural selection, as is any morphological or physiologic trait. Evolutionary genetics also studies the mechanism that results in the changes in allele frequencies in population over time and the consequences of those changes. Okay, so we have discussed about this, and the Hardy-Weinberg equilibrium can also detect whether an entire population contains mutation, genetic drifts, migration, natural selection, or random mating or breeding. So these are the important terminologies for you to take note of. Now that ends my lecture on the introduction to cytogenetics. So this is a relatively easy topic. You just have to read and know the important facts and the history of cytogenetics and how it came to be, and as well as the fields that is the fields of genetics that is highly important for us to appreciate our evolution. Thank you.